Hello everyone, I am Nivedita Jena from Ambika Prasad Research Foundation. Welcome to all of you to our channel Biodiversity and Conservation. So today we are going to discuss about the chromatography and its techniques, types and its principle. So let's start. First we have to know that what is chromatography. Chromatography is the technique for the separation, purification and testing of compounds. The term chromatography is derived from Greek, in which chroma means color and graphene means to write. So in this process, we apply the mixture to be separated on a stationary phase, or we can say that solid or liquid phase, and a pure solvent such as water or any gas is allowed to move slowly over the stationary phase, carrying the components separately as per their solubility in the pure solvent. So coming to the principles of chromatography and how it works. In all chromatography, there is a mobile phase and a stationary phase. The stationary phase is the phase that does not move, and the mobile phase is the phase that does move. The mobile phase moves through the stationary phase, picking up the compounds to be tested. As the mobile phase continues to travel through the stationary phase, it takes the compounds with it. At different points in the stationary phase, the different components of the compound are going to be absorbed and are going to stop moving with mobile phase. This is how the results of any chromatography are gotten from the point at which the different components of the compound stop moving and separate from the other components. Um, so, in paper and thin layer chromatography, the mobile phase is the solvent. The stationary phase in paper chromatography is the strip or piece of paper that is placed in the solvent. In thin layer chromatography, the stationary phase is the thin layer cell. Both these kinds of chromatography use capillary action to move the solvent through the stationary phase. The chromatography is a separation method where the analyte is combined within a liquid or gaseous mobile phase, which is pumped through a stationary phase. Usually, one phase is hydrophilic and the other is lipophilic. The components of the analyte interact differently with these two phases. Depending on their polarity, they spend more or less time interacting with the stationary phase and are thus retarded to a greater or lesser extent. This leads to the separation of the different components present in the sample. Each sample component eluates from this stationary phase at a specific time called as retention time, and as the components pass through the detector, their signal is recorded and plotted in the form of a chromatogram. Then coming to its types, there are four major types of chromatography. These are including adjustment chromatography, thin layer chromatography, column chromatography, and the last one is partition chromatography. So the first one is adjustment chromatography. This is the type of liquid chromatography in which chemicals are retained based on their adsorption and desorption at the surface of the support, which also act as a stationary phase. Um, this method is also sometimes referred to as liquid solid chromatography. Retention in this method is based on the computation of the analyte with molecules of the mobile phase which both bind to the surface of the support. The degree of a chemical's retention in adsorption chromatography will depend on the binding strength of this chemical to the support and the surface area of the support, the amount of mobile phase displaced from the support by the chemical, and the binding strength of the mobile phase to the support. In electrostatic interactions, hydrogen bonding, dipole interactions, and dispersive interactions, for example, that is van der Waals forces, all may affect retention in this type of chromatography. And the binding strength of the mobile phase With the support in adsorption, chromatography is described by the mobile phase's eutropic strength. A liquid or solution that has a large allotropic strength for a given support will act as a strong mobile phase for that material because this mobile phase will tend to bind tightly to the support and cause the analyte to elute more quickly as it spends more time in the mobile phase. As an example, a relatively polar solvent such as methanol will have a higher allotropic strength for a polar support such as silica than a non-polar solvent such as carbon tetrachloride. In the same manner, a liquid or solution that has just low allotropic strength for a support would represent a weak mobile phase for that support in adsorption chromatography. Three types of adsorbents are generally used in adsorption chromatography. One is polar acidic supports, the second one is polar basic supports, and the third one is non-polar supports. The most common polar and acidic support used in adsorption chromatography is silica. Then coming to the second one, which is thin layer chromatography or TLC, we can say in this process of thin layer chromatography, the mixture of substances is separated into its components with the help of a glass plate coated with a very thin layer of adsorbent such as silica gel and alumina as shown in this figure. The plate used for this process is known as chrome plate. The solution of the mixture to be separated is applied at a small spot at a distance of 2 cm above one end of the plate, and the plate is then placed in a closed jar containing a fluid termed as an eluent, which then rises on the plate carrying different components of the mixture at different heights. The rate at which each compound in the mixture moves up the plate depends on its relative attractions to the stationary and mobile phase. Then the third one is column chromatography. It is the technique used to separate the components of a mixture using a column or suitable adsorbent packed in a glass tube, as shown in this figure. And the mixture is placed on the top of the column and an appropriate event is made to flow down the column slowly. Depending upon the degree of adsorption of the components on the wall adjoint column, the separation of the components takes place, and the component with the highest absorptivity is retained at the top, while the other flow down to different heights accordingly. And the five major chromatography methods that use columns are gas chromatography, liquid chromatography, ion exchange chromatography, size exclusion chromatography, and chiral chromatography, etc. Then the partition chromatography. The partition chromatography principle states that a solute will distribute itself between two immiscible liquids according to its distribution coefficient. The higher the distribution coefficient, the more the solute will partition into the more concentration liquid. So in this process, a continuous differential partitioning of components of a mixture into stationary phase and mobile phase takes place. The example of partition chromatography can be seen in paper chromatography. In this process, chromatography paper is used as a stationary phase, which is suspended in a mixture of solvents that act as a mobile phase. And here we put a spot at the base of the chromatography paper with a mixture to be separated, and as the solvent rises up this paper, the components are carried out different degrees depending upon their retention on the paper. The components are thus separated at different heights. So here are the applications of chromatography. In bioanalytical chemistry, chromatography is mainly used for the separation, isolation, and purification of proteins from complex sample matrices. And in cells, for example, proteins occur alongside numerous other compounds such as lipids and nucleic acids. In order to be analyzed, these proteins must be separated from all the other cell components. Then the proteins of interest might have to be isolated from other proteins and purified further. And chromatography is an essential part of almost any protein purification strategy. A number of different chromatography techniques are used for the purification and analysis of proteins. They can be classified according to the physical principle involved in the separation process, and typical examples include reversed phase chromatography, ion exchange chromatography, affinity chromatography, and size exclusion chromatography. And this chromatography is a method that is used in laboratories for the separation of mixture. It is used to test drug levels and water purity. It is also used to determine the nutritional value of the food sample, and it is used to determine the type of chlorophyll in various photosynthetic organisms. And additionally, in chemical industries, forensic science, in environmental analysis, and also in law enforcement, it can be used.